Hi, I'm Dr. K.S. Jyoti. In this video, let us understand a very important molecular mechanism, protein synthesis, what is called as translation. Now, we've understood in our previous sessions that the central dogma of life talks about the flow of information in the or the important molecular processes that are associated with life. So, we started with DNA, which undergoes a self-replication process and is transcribed into RNA molecules in the process of transcription, and this RNA that is a product of transcription now is translated to give rise to proteins. We understood that there is a specific genetic code which relates to your transcription and translation processes. So, the language of nucleic acids is converted into the language of proteins or amino acids with the help of this genetic code. So, genetic code is a triplet codon where 3 nucleotides together will code for one amino acid. So, all the 3 types of RNA, mRNA, rRNA and tRNA are associated together in the process of translation. Now, like any other molecular process, even the process of translation takes place as 3 major steps: initiation, elongation and termination. But before the process of translation can get initiated, first the tRNA molecules have to be charged, or what we call it as acylated, so that the tRNA knows which amino acid it has to bring to the site of protein synthesis. So, we will now understand what are the different steps in the process of translation and how the mechanism is carried out. So, the first step is called as amino acylation of tRNA, or we can simply call it as charging of the tRNA molecule. Next is initiation, elongation, termination, and in eukaryotes generally, for the protein to become functional, it also has to undergo post-translational modifications before the protein can be targeted to a specific organelle where it has to perform its role. So, let us now understand how the charging of the tRNA molecule takes place, or amino acylation of tRNA takes place. So, in amino acylation, the tRNA is combined with the amino acid, and in an ATP dependent process, this reaction is catalyzed by enzymes called as amino acyl tRNA synthetases. So, in this, the tRNA is activated with the help of an ATP molecule and is combined with its specific tRNA, giving rise to amino acyl tRNAs. That means, for example, let's say there is a tRNA which is specific to be bound to methionine. Now, methionine is coded by the codon AUG. So, AUG is the codon for the amino acid methionine. Now, its complementary anti-codon will be CAU. So, the tRNA which has to bind to methionine will have an anticodon CAU. So, the CAU now will read AUG codon. It will read the codon AUG. So, on the 3' end to the adenine, there will be a methionine attached. So, this will now become methionine tRNA. So, like this, all the 20 standard amino acids which are coded by specific codons will be bound to their tRNAs, and they will be ready to be brought to the site of protein synthesis when the process begins. So, the first step before the protein synthesis can begin is amino acylation of tRNAs, or charging of tRNAs, which is carried out by the enzymes amino acyl tRNA synthetases. Now, there is an important point that we have to remember here. Now, unlike in replication, there is no proofreading that takes place in translation. That is, there is no other system which will come and cross-check whether a tRNA is bringing a right amino acid to the site of protein synthesis or no. So, the entire responsibility of attaching a right amino acid to the right tRNA rests with amino acyl tRNA synthetases. So, they have a very responsible role to play in the process of translation. So, they ensure that the tRNA is bound to its corresponding amino acid only. And in situations where there is one amino acid which is coded by more than one codon, for example, let's say we have serine which has about 6 codons. So, all the 6 codons coding for serine will be identified by the same tRNA, which we call it as a sterile tRNA. So there will be only one tRNA bringing serine, but whatever codon codes for that will be identified by the same tRNA molecule. Now, how is this made possible? Now, here we can see when we look at a codon when we are reading, now let's say this is 1, 2, 3. Now, the alignment in an anticodon is in the opposite direction. This is 1 prime, 2 prime, 3 prime. This is your anticodon end. Now, it is observed that the third position of the codon or the first position of the anticodon is mostly occupied by what is called as a Wobble base. Wobble base like inosine, which is an unusual base. Now, inosine can base pair with adenine, uracil and cytosine. So, any codon which is having ending with any of these three for the same amino acid can be identified by the same anticodon end of the tRNA. So, this Wobble hypothesis was put forward by again Watson and Crick to actually explain how redundancy in genetic code can be, or degeneracy in genetic code can be accepted. That is, amino acid, even if it is coded by more than one codon, there is only one tRNA which can accept any of these and bring the same amino acid to the site of protein synthesis. So, Wobble hypothesis is a very interesting phenomenon using which the tRNA's anticodon can identify any codon coding for the same amino acid. So, the first step is we are making all the tRNAs 20 tRNAs to be ready with their respective amino acids to be ready to bring them during the site of protein synthesis.